Why test a piece of equipment for cleanability? EHEDG develops and publishes practical guidelines for hygienic design of equipment used in food processing. That is, general criteria, specific requirements for various equipment types, and recommendations for equipment used in various food sectors. However, if for functional reasons it is not possible to fully comply with the guidelines, and in any case of doubt, it is necessary to test a piece of equipment to verify its cleanability. EHEDG has developed a particularly recognized method to test cleanability of closed pieces of equipment, such as valves, pumps, sensors, and others. This method is described in the EHEDG Guideline Number 2. Principles of the method. First of all, the EHEDG Document 2 method is a cleanability test. This means that it is intended to experimentally examine a piece of equipment with regard to its cleanability, using a standardized soiling and cleaning procedure. It is a screening test for hygienic equipment design, which allows revealing critical areas. It should be noted that whilst this method is an indicator of good inherent hygienic equipment design with respect to in-place cleanability, the onus is still on the user to verify the cleaning performances in the industrial situation with the real product in use. The EHEDG Document 2 method is a comparative test. The test item is examined in comparison with a straight piece of stainless steel pipe with controlled roughness, called the reference pipe. In order to ensure adequate repeatability, all authorized laboratories use standardized reference pipes obtained by the same raw materials and manufacturer. In the EHEDG Document 2 method, the cleanability is evaluated at a microbial scale, whose size is in the order of the micrometer. The test item is soiled with a soil containing microbial spores. The remaining spores are detected after the CIP. How does the detection of remaining spores work? Since spores are very small, it is impractical or even impossible to see residual spores on the surfaces. To make residual spores detectable, they are allowed to germinate and grow in a specific medium, which is put in contact with the surfaces after CIP and then incubated at a specific temperature. The microbial growth provokes a discoloration of the growing medium from purple to yellow, allowing easy detection of any difficult to clean area. Steps of the method. Prior to testing, all gaskets and polymeric materials must be checked for antimicrobial properties. The materials must not interfere with the microbial growth in order to avoid false negative results. This is obtained pouring inoculated agar on the samples of the polymeric materials. If a purple zone is seen around the samples after incubation, this means that the material has antimicrobial properties and thus can't be used in the cleanability test. Prior to testing, the test item, the reference pipe, and the ancillary fittings are dismantled and thoroughly cleaned and sterilized. Sterilization can be achieved by autoclaving, inline steam steaming, or by chemical decontamination using a suitable biocide. The test item is then reassembled and coupled to a 250 mm long reference pipe with two short auxiliary lengths of pipe at each end to form a test section. The reference pipe, auxiliary pipes, and inlet to the test item should all be of the same internal diameter. A sour milk inoculated with approximately 10 to the power of 5 to 10 to the power of 6 Geobacillus stereothermophilus spores per milliliter is used as the test soiling medium. Geobacillus stereothermophilus is a thermophilic microorganism that doesn't grow at normal ambient temperature, thus allowing sample handling in non-sterile conditions. The test section is filled with the inoculated sour milk and closed at either end. The test section is then pressurized 3 times to 5 bar, using a closure plate with a suitable air union, and held at pressure for 2 minutes on each occasion. Whilst under pressure, any movable parts are operated to simulate in-use conditions. The sour milk is then drained. A sample is taken and pour plated to determine the actual spore's concentration. This is done at this stage instead of prior to soiling, to account for any dilution of the milk soil with water retained in the test item after autoclaving. The test section is connected with a suitable air union and dried by flushing air with dry filtered air at a mean velocity of approximately 1 meter per second in the reference pipe. This may generally take between 2 and 4 hours, depending on the internal area of the test item. After drying, the soil test section is mounted in a purpose-built test rig. The following cleaning procedure is then initiated. A pre-rinse with cold water, a cleaning phase with a mild detergent solution at 63 degrees for 10 minutes. A final rinse with cold water. For all pipe sizes, cleaning solutions are circulated at a mean velocity of flow of 1.5 meters per second. Samples of the two rinse waters are taken and the presence of spores is determined by pour plating with a specific growing medium. After cleaning, the test section is removed from the rig. The internal surface of the reference and the relevant internal surfaces of the test equipment are covered with a molten growing medium called MSH agar to a thickness of at least 5 millimeters. After the agar has fully solidified, the reference pipe and the test item are placed in an incubator at 58 degrees for 16 to 24 hours. At this temperature, the remaining spores will germinate and allow for discoloration of the agar from purple to yellow. After incubation, the test equipment and reference pipe are examined for the presence of areas of yellow discoloration in the purple MSH agar. A counting grid with 5 by 5 mm graduations is used to facilitate the assessment of yellow areas. Experience has shown that a yellow area between approximately 5 and 30% for the reference pipe is optimal for a good interpretation of results. Result interpretation. Assuming that the extent of the yellow area in the reference pipe complies with the acceptability range, 4 results are possible for the test item. 1. If visible milk residues are present in the test item, there are serious hygienic design faults inherent within the test item. The test should be repeated, and if milk residues are found again in the same area, no further tests are required and design changes should be stressed to the test item manufacturer. 2. If yellow areas are present in the test item, the test procedure is repeated from 3 to 5 times to investigate whether the yellow zones are randomly distributed or indicative of poor hygienic design. Presence of yellow discoloration in the same area of the test item on 3 separate occasions is indicative of areas that are difficult to clean, and hence an improvement should be considered. If yellow areas are randomly distributed, the method doesn't reveal critical areas with regard to hygienic design, and the cleanability of the test item can be compared with that of the reference pipe based on the percentage of yellow areas. 3. If no yellow zones are present in the test item on 3 successive occasions, no further test repeats are required, and the test item can be described as particularly cleanable. The EHEDG Document 2 is also used by the EHEDG authorized for the EHEDG Type EL Class 1 certification. For further details, please refer to the EHEDG website www.ehedg.org.